do attendance. Again, I'll just call out your name. I don't think you're here. So if I call out your name and you're here, just let me know. As far as I know, only one person has WVSU gear on, right? Okay. So if you want to do extra credit, take a picture at the end for even more extra credit, let me know. And then obviously we'll need a volunteer to take our picture too for extra credit. We'll cross that bridge when we get there. All right, Carr, you're back there, right? Carr, sorry, last name?
Davis.
That's fair. Gotcha. Okay. So Carr is not here. Smith, Newton, Stanley, Fisher, Sims, and Wazell. Right, so I didn't miss anybody. All right, before we get started, I'm sure some of you guys have noticed, and I apologize, but I am getting a little bit frustrated, and that's my fault. I'll explain why in the context of lab, for the most part. I see this every semester. A lot of people are doing poorly, and I'm like begging you to do better. Like, hey, you can make these corrections. Hey, you didn't do this assignment. You can still do this assignment. And I'm saying it and saying it and saying it. And then a lot of people are just ignoring it. And I know what's going to happen at the end of the semester. Most people won't make any changes. And at the end of the day, what can I do about my grade? I've got a scholarship and I can't fail. And I'm telling you now, this is what you need to do about your grade. Make the corrections. Do the assignments that you've missed. Come to me for help. Take it seriously, right? These are the things you need to do. And at the end of the semester, like I said, some of you are going to come to me and say, I need to do something about my grade. And I'm going to say, there's nothing you can do about it because I've been begging you all semester. And then you guys are going to do the student evaluations and I'm going to get poor grades because you're going to say I'm not fair and things like that. I've seen it happen. Actually, my numbers have been going down since COVID. And since COVID is when I started doing this thing where I'm like, it used to be if you were late, and that's it. Whatever you turned in, that was it. There was no correcting it. But since COVID, I've been getting more flexible. But I think because of me getting more flexible, then I'm also more like bothering you guys about it. And I think that pisses people off. And then at the end, when they ask for help, and I'm just like, nope, I've been telling you all semester, then they get pissy about it. So it's actually been hurting my numbers, but I'm fine with getting hurt numbers as long as it's good for the students. As much as you might be irritated with me, this is better for you. So please get to work. Please do the stuff you've missed. Please make your corrections. Please come to me for help. Speaking of that, I need a volunteer. You don't have to volunteer now, but if someone can meet me online for office hours today, I need to make a short video from your end that shows what it looks like on the student's end to go into your labs and see the comments I've left. Because I got an email today from someone who didn't know that I'd left comments, which is understandable because this thing, Google Calendar or Google Classroom, can get a little confusing. So I want to make sure that you all know how to go in and see the comments I've left, because you can't fix mistakes if you don't know which mistakes you made. So I want to help you. And please come to me for help. Like I said, as of last week, you'll get extra credit just for visiting me during office hours.
That's every day.
Well, once a week, yeah. I mean, you can visit me all the time, but yeah, you'll get the points once a week. Just for stopping in, and if you're doing great, it'll be a quick meeting. I'll just be like, you're doing great. There's nothing much I can tell you. Or maybe you can go help your classmates. So yeah, now's the time. We're almost midpoint. So now's the time to fix your grades. And I am here to help. And I am very flexible until at the end. At the end, I won't be flexible because I'll say, I've been telling you all semester to do this. Anyway, any questions about that?
Can I say something?
Please.
My independent work sometimes is more like a conversation with him. Like it's not just, oh, this scientific fact. It's more like, like I'm talking to him. He's just not responding. So it's very relaxed.
Thank you, yeah. And that's
If that helps.
Or he's not talking the whole way.
Right, like it's more like me telling a story about what's happening. So it's not like really, really rigid and strict. If that makes sense. I mean, you gotta cite your stuff, but just.
Yeah, independent work is great for that reason. It's not just for me to grade you. Sometimes it is. If I'm really busy, I'll just give you a grade. But sometimes it's a good learning moment, as some of you in this room know. And I'm happy about it because I love those interactions. I'm like, you did lose points for this, but this source you posted is really biased. So that's a great learning thing. Like keep that in mind in real life, not in biology 101, but in real life. It's a great way to learn stuff. That's what I like about independent work. Sometimes it is just as simple as turning in a paper. But a lot of times it allows us to, like you said, basically go back and forth and learn. So yeah, thank you. And I want to call it free 100 points because you have to work to get the 100 points, but it is free in that it's completely in your control, right? It's not like you're getting tested on anything. All you have to do is turn in work. So
Can you go over 100 points?
yes, good point. Anything over 100 points is just extra credit. So 100 points is not the ceiling. 100 points is the perfect score, and anything beyond that, extra credit. Great question. Any other questions? Yes, sir.
When you're doing like independent work, can you like give your own opinion too?
Yes, definitely. Opinions are good, yeah. Especially if you can back it up with some sources. And that would be a great, again, you'll see if you do it, it'll be a great starting point for a conversation. But yeah, this source does support your opinion, and I'll come back and say that again, but you might want to consider this source. It goes against your opinion. And you won't lose points for that. It'll just be a good conversation.
Okay, I was thank you.
Yeah. Problem. Any other questions? I love these questions today. Very good. All right, so then we'll get back down to business, I guess, the boring stuff. I guess the first word for attendance, let's go with Calvin, since it's on the screen there. Calvin is the first word for attendance. Pull it up here. And Calvin, first word for attendance. Okay. Anyway, to remind you, we are in chapter 7, which is the last of the 3 chapters that will be on the second exam. The first chapter we covered was chapter 5, and there's a lot of stuff you need to know, but if anything else, just remember, so your cells need ATP to live, right? ATP is the form of energy that your cells use, which begs the question, where does the ATP come from? And that was answered in chapter 6, which the answer is cellular respiration. That's how we make ATP. We make ATP with the process of cellular respiration, which happens to break down glucose, which brings up the next question. Where do we get glucose? And that's the answer to this. This chapter gives us that answer. Chapter 7, photosynthesis is how we get glucose. And to remind you, we've already kind of done an overview of photosynthesis. And now before we get into the details of photosynthesis, we're kind of learning a little bit of background information that you need to know. So for example, we talked about the nature of sunlight. For example, my shirt right now is yellow or gold, depending on what you want to call it, because it's absorbing every single color other than yellow. So that's the color that's being reflected, right? As far as the lab that we're going to do today, that's the most important information from that bullet point. We talked about chloroplast pigments. There's 3 of them that we talked about, chloroplast A, chloroplast B, and the carotenoids. And I said you don't really need to distinguish between the 3. It was just for your own information. And now we're getting into this, how the photosystems harvest light energy. So let's talk specifically how it works. As you already know, and I'm going to put it next to this because you don't need to know this for the exam, light behaves as waves, right? Because I talked briefly about how like the shorter the wavelength, the more energy there is, which is why blue light is more energetic than red light. But it also behaves as discrete packets of energy called photons. So even though I put it next to that, I will circle this because you do need to know what a photon is. I will use this word a lot. A photon is a fixed quantity of light energy. Basically, so when I say photons, I'm saying, how do I say this without being incorrect? All right, we're not talking, this isn't a physics discussion. So as far as what you need to know in this class, you could almost think of photons as electrons, right? Electrons are like, that's, the negatively charged thing in atoms, right? It's a form of energy, right? And in a way, photons are the same. It's a form of energy, except we're not talking about atomic energy, we're talking about light energy. So when I say photons, we're just talking about light energy. And in a way, you could almost think of it as this little thing, little tiny things that we can't see, like an electron, we can't see the little things coming at us from a light source. Anyway, that's what a photon is. It's not that, photons can be really complicated if you really study them, but in the context of this conversation, they are not that complicated. And again, like I said earlier, a few seconds ago, the shorter the wavelength, the greater the energy of the photon. So I'm going to put it next to that too. Because for this course, that doesn't really matter. And think about it, we had this discussion on Monday. I mentioned, as far as we're concerned, for that lab you're doing today, when we're talking about the color, like which color is worse for photosynthesis, which color is best for photosynthesis, really the only thing we're concerned about is what color is being reflected and what color is being absorbed. So we know the green light is being reflected. Therefore, that's the one color it can absorb. Therefore, hypothetically or theoretically, based on what you know, green light should be the worst, based on the information I've given you. And also, on the same note, we know that white light is all the lights, therefore we would assume that white light should be the best light. But yeah, we didn't specifically talk about the energy of the photon. Some of you did, and I was impressed with that. For your pre-lab, when you did your hypothesis, and you said, I think this color will be better because it has a higher energy, which was very good. I hadn't even taught you that yet. Anyway, any questions so far? All right, there's a lot of information here. But when a pigment, we can go through this quickly because half of this is not new. But when a pigment molecule absorbs a photon, the pigment electron gains energy. So, again, if you've ever worn a black shirt on a sunny day and you got warm, that's why. It's because the photons are hitting that black shirt and black absorbs all the colors. So all the electrons, literally the electrons that are in the molecules that make up whatever pigment is in your shirt, or if we're talking about chlorophyll, the electrons in the chlorophyll pigment, it gains electricity, or excuse me, it gains energy. Which is important because remember, when we were talking about cellular respiration, I kept using the words high energy electrons. I didn't say electrons, I said high energy electrons. We were stripping away high energy electrons from glucose. We were stripping away high energy electrons from pyruvate. We were stripping away high energy electrons from citric acid, right? Stripping away high energy electrons. So that's where they get their energy. Ultimately, they're getting their energy from the sun. The sun hits it, it excites it. But when electrons are excited, it's a very unstable state, which is a very big statement when you talk about physics and really get into it, but we're not getting into it. Let's just say it gets excited and it can't really stay excited. So what happens is it usually loses its excess energy and falls back down to the non-excited state, like right away. But again, that's why you get hot when you're wearing a black shirt outside, when there's a lot of sun. Because those electrons literally get excited. Energy can't be created or destroyed. So when it loses that energy has to go somewhere, right? So in that case, that energy releases heat energy. Of course, as far as we're concerned, for photosynthesis, this bullet point doesn't matter. So yes, for your everyday life, that matters. Again, when you're wearing a black shirt, that's how you get hot, because those electrons get excited and they can't stay that way, so they lose that energy. And that energy goes and it's released as heat. That's why you get hot. But for photosynthesis, we couldn't care less about that. Because photosynthesis isn't about getting heat energy, right? It's about getting sun energy and turning it into chemical energy. So that's why I put it next to that. It's interesting to know that, but you don't need to know it to understand photosynthesis. But everything else above that, you definitely need to know. So pigment, again, gets hit by a photon, and the electrons in the pigment get excited. That's very unstable. So actually, let's say this right here. From there up, that's the most important as far as the exam is concerned. Photons hit pigments, which excite electrons, and now the electrons are excited. Yes, this other stuff is true. The photosynthesis is going to intervene. The process is going to intervene. Once those things get excited, photosynthesis is going to take advantage of that. The next item I'm going to show you is a picture of this. Are there any questions while I have the words on the screen? Okay, here we go. It's basically a picture of what I've shown you, right? So there's a chlorophyll molecule. It's just like any of the other molecules that you've drawn for lab 2. Remember, you drew molecules. Same thing, right? It's just a bunch of atoms bonded together that have electrons in between them. So then they get hit with the light, they get hit with the photons, that excites them. And what happens, and you know what, I'm gonna put it next to this too, because this is just from your book, but yes, typically what happens is they get excited and they can't stay that way, so they lose their energy. And that energy is then released as heat. Sometimes it's released as light, like things that glow. That's why they glow. But again, photosynthesis, this is pretty much where it stops. Well, not stops, but it doesn't usually go here. Photosynthesis takes advantage of that excited electron. We'll talk about how shortly. I don't even know why your book talks about this next concept, but your book did, so will I. So bear with me. Sometimes when these electrons get excited, like I said in that last picture, and then they lose that excitement, like we said, sometimes they release heat, but sometimes they also emit light. I'm gonna put a big old X to this. I don't know why your book thinks you need to know this. You can forget this information and be fine for the rest of the semester. You can forget this information and completely understand photosynthesis. So I don't know why they included it. I guess it's kind of neat. So what happens is, I'm sure you guys have seen glow sticks before, right? So what happens in that case is, you break them, and then there's two different chemicals, and they mix. And what happens is that excites the electrons in those chemicals. Of course, it's not getting excited by light. It's kind of the opposite. Those things get excited. They can't stay that way, so they lose their excitement. They lose that energy, and they put it off as light. So anyway, any questions about that? Completely not important for what we're talking about. So let's go back to what is important. In the thylakoid membrane, remember that's the part inside the chloroplast where photosynthesis is happening. The chlorophyll molecules are organized with other molecules into something called photosystems. I want to point out that I'm not going to ask you what a photosystem is. I'm just going to use the term photosystem when I'm explaining how photosynthesis works. So that's the reason you need to know what a photosystem is, just so you know what I'm talking about when I use the word. I'll show you a picture soon to show you what a photosystem is, but for now, I'll just describe it verbally. Each photosystem has a cluster of a few hundred pigment molecules. I'll definitely put it next to that. That's good for you to know, I guess, but it doesn't really matter. You don't need to know those numbers. Here it is. There's a whole big chloroplast with all the thylakoid membranes inside. Let's zoom into the chloroplast and zoom into that particular thylakoid membrane, right? It's the phospholipid bilayer. We zoom in, and here's just a big chunk of something. And that big chunk of something, that is a photosystem. And the photosystem has other stuff, but more importantly, most importantly, it has a bunch of pigment molecules. So it's got some chlorophyll A, got some chlorophyll B, got some lycopene and beta carotene and all those things we talked about at the end of the class on Wednesday. And that's where it all is. So that's what a photosystem is. You could almost think of one. Yeah, I don't want to say that. It's just something embedded right there in the membrane of the thylakoid membrane. And I'm still going to give you some more information about these photosystems. But are there any questions so far? Excuse me. All right. Phew. So this cluster that we just saw, and I'm going to show you another picture of it soon that has more information on it. It's kind of like a light gathering antenna. That's how your book describes it. I would describe it, I think it would be more accurate or more relevant. You could almost think of it like a solar panel. Because you could really think of chloroplast as little factories. What are the factories making? They're making glucose. Just like any factory, they need ingredients and they need energy to make stuff. Where are they getting their energy? From these solar panels, right? From the sun. So what happens? We already know this, but we're saying it again. The photons from the light, usually the sun, but whatever, from any kind of light, strikes the pigment. The electrons get excited, but instead of like going back down and release this heat or light like we talked about earlier, this time the energy just kind of jumps around from molecule to molecule until finally it goes to the reaction center. As far as I'm concerned, we could just simplify things. You could almost forget that whole point. I mean, yeah, it's true. It's not like the photon hits the pigment, the electron gets excited, and it goes directly to the reaction center. That's not what happens, but as far as we're concerned, it might as well be. The fact that it jumps around from molecule to molecule isn't very important for you understanding how photosynthesis works. I'm going to show you a picture of what the reaction center is. So for now, just know it as a word. Know that the reaction center consists of, actually, you know what? You don't even need to know that. Yes, technically, the reaction center does consist of chlorophyll A. Because remember, there was chlorophyll A and chlorophyll B. And yes, technically, chlorophyll A is the one in the middle of the reaction center, but you don't even need to know those details. What you do need to know is that right there in the reaction center is this thing called a primary electron acceptor, which is important because remember, right now, this is all about getting those excited electrons. So the light hits the pigment. The electron gets excited. It bounces around until it hits the reaction center where finally something grabs it, so to speak. Something is grabbing that high energy electron while it's still high energy before it goes down and loses its energy. That thing is called the primary electron acceptor. Most likely, I will not ask you what a primary electron acceptor is on the exam. But you need to know what it is because I will use that term. So when I say primary electron acceptor, you'll know what I'm talking about. That's why it's important. It's almost, now you said I don't want to confuse it. I won't confuse it that way. Just remember, again, the electrons get excited from the solar energy, from the light energy. And then this thing, the primary electron acceptor, is the thing that gets it while it's in its excited state. And then another team of molecules uses that trapped energy to make ATP and NADPH. And that was already something we covered Monday when we did an overview of photosynthesis. And that was 2 test questions. And I've already covered it, but let's say it again. Which of the 2 stages produces ATP? And the answer is the light reactions. Which of the 2 stages produces high energy electrons in the form of NADPH? And the answer is the light reactions. Because remember, the light reactions are all about making the energy, not making it, because you can't create it or destroy it. But again, you could think of chloroplasts as little factories that make glucose. And it's the Calvin cycle, that's the actual factory that's doing the glucose. This part, the light systems, those are just the part getting the energy to pump to the factory. So anyway, the next slide I'm going to show you is basically a picture of all these words. But are there any questions about the picture, the words that are on there right now? All right, here's the picture then. Again, we've already seen this left half of the picture. We already saw the chloroplast. We zoom into this thylakoid membrane here. We can see the photosystem that's embedded into the membrane. Now we're going to zoom in, kind of get a bird's eye view of this photosystem, the top of it. So you can see all these pigment molecules. And like I said earlier, the photon hits the pigment molecule. It excites the electron. That electron just bounces around. The excited electron just kind of bounces around for a while until it finally hits the middle, which is the primary electron acceptor. And that's the thing that gets that electron when it's in its high energy state, because that's what we're after. The 2 things we're after for the light reactions are high energy electrons and ATP, right? So we're going to need, obviously, these high energy electrons. We need to get them while they are high energy. And again, I'll put some X's through here to remind you that you don't need to know it's chlorophyll A. You just need to know that eventually that excited electron gets captured by the primary electron acceptor. Which maybe that'd be a good extra credit question. You know, which chlorophyll molecule captures or finally gets the excited electron? And that would be chlorophyll. You could almost think of it like chlorophyll A is obviously the primary, that's the important one, right? It sounds like chlorophyll A would be important, chlorophyll B would be less important, so that'd be a way to remember it. Not that you need to. Anyway, any questions about this picture? All right, here's the next word for attendance. I'm going to underline it. Energy is the next word for attendance. If there's no questions about this slide, we can move on to the next PowerPoint. So we know now how the light reactions, or the photosystems in the light reactions, how they harvest that light energy. We know it now, right? Because obviously you can't go directly from light energy to chemical energy. So we know the first step, which is basically to use light energy to excite electrons and gather those excited electrons, right? We need to get them while they're excited. So that's what that last bullet point was, right? The photons excite the electrons and we capture them while we're excited. Now we'll move on to the next step. And we're still talking about light reactions because we still haven't said, how do we make ATP? How do we make the NADPH? So let's talk about it. So we talked about photosystems, right? That's the last thing we talked about. Now your book goes on to get specific and says, there are 2 photosystems that cooperate in the light reactions. Yeah, I guess you do need to know that. Not that I'm going to ask you how many photosystems there are, but you do need to know this happens in 2 stages. So you'll see, and hopefully it'll be intuitive as we go through it. So what happens is the photons excite the electrons in the chlorophyll of the first photosystem. And again, the number doesn't matter. It really doesn't matter, because if you were to watch that video I posted with that short video from that guy named Hank, where he talks about it, it's actually a little bit confusing. Because the way they talk about it, it's the second photosystem is talked about first, and then the first photosystem is talked about second. So I'm going to leave numbers out of this. Just know that in one of the photosystems, the first one that we're talking about in the process anyway, the photons excite the electrons, right? So that's the one we just talked about. That's the one I just showed you. Photons hit the pigments, that electron bounces around, it's all high energy, and then finally it gets to that primary electron acceptor, and that thing catches it while it's high energy, right? That's the thing we just talked about. Which is up to this. That's the point. We've already talked about that. From here up is what we already talked about in the last main bullet point. Then what happens? Well, electrons have to come from somewhere, they have to go somewhere. So if we just took an electron out of that system, then we have to replace it. That's what this next photosystem, that's what this next bullet point is. That electron that was just taken away because it got excited and went to the primary electron acceptor, it's gone now, right? So it has to be replaced. Where does it come from? It comes from the water. And I already mentioned that on Monday, so that, again, that's a good test question. Which of the two stages of photosynthesis uses water? The answer is the light reactions. Or where do the electrons come from in photosynthesis? Because that's a separate test question. And the answer is water. And what happens once that water gives up that electron? And the answer is it becomes, basically, as far as we're concerned, it becomes oxygen. Obviously, H2O isn't just going to magically turn into O2. But as far as we're concerned, we don't need to worry about those details. Just know that water says, hey, here's an electron. And when that happens, that produces oxygen. So there's another test question. Which of the two stages of photosynthesis produces oxygen? And the answer is light reactions. And I don't know if I said this before or not, but this is a good time to remind you. It's almost like a trifecta, a triangle. For this exam, you should think of these things together. Oops. Water, electrons, and oxygen, right? Because in respiration, which is what we talked about in the last chapter, oxygen accepts the electrons and becomes water, right? In photosynthesis, water gives up the electrons and becomes oxygen. So no matter what we're talking about in this chapter, in this exam, whether we're talking about chapter 6 or 7, whether we're talking about respiration and photosynthesis, these things are going to go together, right? Oxygen, water, and electrons will go together in that context. The next slide is going to be a picture of the words that are on this slide. But are there any questions about the words that are on this slide? All right. So here we go. It's basically sort of like a picture of what you saw earlier, just slightly different. This part's pretty much the same. I mean, you already saw this picture, except last time I showed you, it showed you the thing bouncing around a lot, and then it showed you hitting chlorophyll A. But this time, I like this picture better. This is what you need to know. So this is the same thing I showed you earlier, but without all those other details. This is what you need to know, right? Light hits it, it excites the electrons. That excited electron is eventually captured by the primary electron acceptor. I'll also go ahead and point this out and put it next to it. Technically, I keep saying an excited electron. Technically, it's 2 electrons at a time. They kind of move in pairs, but you don't necessarily need to know that. So again, we hit the pigments, we excite the electrons, and when they're in their excited state, the primary electron accepts them in the excited state. Meanwhile, since we took some electrons away and we have to replace them, that's where water comes in. Water says, here you go, here's some regular little electrons. You can use the light to excite them. And in the process, that makes oxygen. And again, I'm going to put it next to these numbers, because we're not getting that detailed. Yes, technically it's 2 electrons. And yes, technically, when the water gives up those electrons, then we get 2 hydrogen ions, and half a molecule of O2, but we're not worried about that. All you need to know is the water gives up the electrons, and that's what gives us oxygen. Which is good, right? Because without that, we would die. So again, like I told you, this whole thing, when we're talking about the light reactions, happens in 2 photosystems. This is one of the photosystems. Now we're talking about the next one. So here we have this high energy electron, and we still haven't done anything with it, right? So let's put that high energy to use. That's the next part. The next photosystem. And again, I'm just going to put it next to that number, just because sometimes it can get confusing. So it doesn't matter, the numbers don't matter. We talked about one, now we're talking about the next one. So again, we have this energized electron. What do we do with it? Well, the energized electrons from the first photosystem passes down an electron transport chain to the second one. There we go. we're back to electron transport chains. The same, as far as we're concerned, in this level of, this 100 level course, it's the same type of electron transport chain that we used in respiration. So if you've already memorized what we learned in respiration, then you know exactly what you need to know here. It's the same thing. The electrons go from protein to protein. They start at high energy, and every time they go down, they release some energy. That energy, just like in respiration, is used to pump hydrogen ions against their concentration gradient, right? Because it takes energy to make something go against its concentration gradient. Then nature does its thing. They want to diffuse down the concentration gradient, so they do. And again, just like in respiration, that goes through the ATP synthase, right? That enzyme that literally turns like a turbine and produces ATP. And that's where we get the ATP. So again, electron gets excited, gets captured by that primary electron acceptor. That's the first photosystem we talked about. Then, on its way to the second photosystem, it goes down an electron transport chain and produces the ATP, which is really important because when you think about it, again, the big picture here, the two things we're after for light reactions are ATP and high energy electrons, right? So there we go. We're halfway done talking about that. We've got the high energy electrons, excuse me, the ATP. Then finally, again, they go down to the electron transport chain. I'm going to show you a picture of this next. They make it to this second photosystem. And keep in mind, too, when they go from the first photosystem to the second one, they're losing their energy, right? Because I kept saying that they were high energy. We captured them when they're high energy. But then I told you they went through the electron transport chain and they released their energy. So by the time they get to the second photosystem, they're not high energy electrons anymore. So what happens? Well, the second photosystem does the same thing. We use light energy to pump them back up and give them more energy. And there it is. There's the whole thing. This is a great picture to study when you're studying for the exam, because it leaves out all the other details you don't need to know. It has the most important stuff. And the risk of being repetitive, I'm going to say it again, so important. The light hits this initial photosystem. It excites the electrons, which are eventually taken away by this primary electron acceptor. And of course, if you're going to take an electron away, you have to replace it. So water says, here's some electrons, which produces oxygen. Then we go back, let's talk about what happens to this excited electron. Since it's excited, we send it down an electron transport chain, right? It goes down, it's releasing its energy. As it does that, we use hydrogen ions. We pump hydrogen ions against the concentration gradient. We let them diffuse back down. That's where we make the ATP. But like I said, we're losing energy. We're losing energy, but then we need to bump them back up, so same thing happens. We have another photosystem, just like this one, right? The light hits it, it excites the electrons. It's then again accepted by a primary electron acceptor, except this time, instead of going back down the electron transport chain, this time the NADPH or NADP plus, that captures the electron. So now at this point, we have everything we need. The two things that we needed from the electron, from the photosystems, from the light reactions are this: ATP and high energy electrons in the form of NADPH. That's it. Now you know the two most important things. That's what we're after, and that's how we get them. Any questions about this slide? So for the exam, I'm not gonna make you differentiate. I won't say something like, is it the first photosystem or the second photosystem that gives us ATP? Is it the first photosystem or the second photosystem that gives us NADPH? You don't need to differentiate between the two. Just know that which of the two stages, light reactions in the Calvin cycle, produces ATP in this light reactions. Which of the two stages produces high energy electrons? And for the exam, I might say high energy electrons, or I might say NADPH. I don't know yet. You should think of those things as the same thing. And the answer is photosystems, light reactions. Any questions about this slide? The next slide I'm going to show you is a picture of the same process, except they kind of use humans. I like this next one. It really makes a lot more sense this way. It's like the same thing, but you can really visualize it better. So here we have this non-energized electron. So we need to energize it with a photon, right? The photon hits it, it bounces up, it has this high energy, which is really kind of accurate too, right? Because remember, you know about potential energy and kinetic energy. Now that it's up here, it has a lot more potential energy than it did right down there, right? So we use kinetic energy to give this thing a lot of potential energy. And now that it's up there, we can just roll it down this ramp, and that's literally going to turn that wheel And just like an ATP synthase, which literally turns and produces ATP, right? So it's going down, it's losing its kinetic energy, or it's losing its potential energy because it's kinetic energy. Then it gets back down here, and it's not enough energy. So boom, it gets hit with another photon, and it gets even more energy. And now that it's highly energized, it gets captured by this NADPH, which I said last week, and this week too, I think. I kept calling it like a bucket that carries around electrons, and that's why. 'Cause I like how your textbook literally illustrates it like that. It's just a bucket that carries around those high energy electrons. So anyway, any questions about this? All right. If that doesn't make sense to you, or if you just want to make sure it does make sense to you, you should watch that short video I posted on Google Classroom, one of those 4. He does a really good job of explaining it. He explains it a different way. He has graphics. He's more entertaining, and it's really short. So I would highly recommend watching that video. And if you want to get a few points for it, you could watch the video while answering the questions. Anyway, this next slide should be really quick and easy because I've been saying it as we went through this, but let's put it in writing so you can read it. The ATP production that we just talked about, right? Using those high energy electrons to make ATP. As far as we're concerned, it's the same thing as it is in respiration. Because in both situations, we have an electron transport chain, which to remind you, is just a bunch of proteins embedded in the membrane. And the electrons go from one protein to the next. That releases energy. That energy is used to pump hydrogen ions across the membrane against the concentration gradient, or up the concentration gradient, or from low concentration to high concentration, right? I said the same thing just now in 3 different ways. Again, we're going against nature, right? It's the opposite of diffusion is what just happened. That's why we needed energy to do it. But the big difference, and this is going to be a test question in a different context, but the big difference is where do the electrons come from? And I keep saying this, I've said it over and over again, but here it is again. If we're talking about respiration, the electrons come from food. Or more specifically, at least for the exam, it comes from glucose, right? We always say glucose just to keep things simple, but yes, technically it comes from everything you eat. But in photosynthesis, it's the opposite, right? So again, respiration, the high energy electrons come from the food. They go down, 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 and finally oxygen accepts it and becomes water. And then you just reverse that for photosynthesis. Photosynthesis, the water says, here's some regular old electrons, and then they get excited, and that's where the electrons come from. The next picture I'm going to show you is a picture of this, a picture of the electron transport chain in photosynthesis as opposed to respiration. But before I show you that, are there any questions here? Okay, the next word for attendance, I'm going to circle it instead of say it. So again, if you're online, relaxing with your eyes closed, listening to the lecture, you do want to open your eyes and look at this part. That is the next word for attendance. Any questions? All right, this should be quick and easy because it's everything I just said, but in a picture. And it should look a lot like what we saw in respiration. Again, here's the first photosystem. Light hits it. It excites electrons. The electrons are eventually accepted by the primary electron acceptor, and it sends it to the next photosystem. But on the way there, it goes down an electron transport chain. It releases its energy. That energy is used to pump hydrogen ions against the concentration gradient. See, there's a lot of them in here, a lot of hydrogen ions in here. There's not a lot out there. So they don't want to go from here to here, right? They want to go from here to here. So we need energy to pump them from here to here, and that's what happens there. Anyway, the electrons, again, they get to the second photosystem. They get excited again because they lost all their energy at the electron transport chain. They get excited again. Then they're finally captured by the NADPH, NADP+. It captures them and takes them off to the Calvin cycle. We produced ATP, which goes off to the Calvin cycle. Yeah, very similar stuff, right? Very similar to respiration. Are there any questions about this? And to jog your memory, the next slide I'm going to show you is what you've already seen. It's respiration. Very similar thing, right? The electrons, these high energy electrons go from, they start with the NADH, and then they're dropped off of the electron transport chain. They go down protein to protein. We're pumping hydrogen ions against the concentration gradient. They don't want to be there, so they diffuse back down, and that's where we make the ATP. So that is the end of photosystems. So remember, photosynthesis consists of two stages, the light reactions and the Calvin cycle. We are done with the light reactions. For your studying, if you don't remember any of the details I just told you, at least remember this. In the light reactions, we produced high energy electrons and we produced ATP. That is the most important thing to remember because that's what the Calvin cycle needs. The Calvin cycle needs energy to squish those carbons together to make glucose. And it needs some high energy electrons to incorporate into that glucose. That way, when we eat a little bit later, we can get those high energy electrons and make ATP. So let's jump into the Calvin cycle. Like I've already said, the Calvin cycle kind of functions as a sugar factory. Well, I kind of said that the whole process is like a sugar factory, right? I kept saying the chloroplast is like a sugar factory. And then you've got two parts of the factory. You've got the part that's actually doing the work, which is the Calvin cycle, and then you have the electron transport chain, which in that analogy would be like the power plant, right? The power plant that runs off of solar energy. So that's important. You do need to know that. This is less important. It's just kind of given. It's called a Calvin cycle. So the fact that it regenerates the starting material with each turn, that's almost a given. Just like the citric acid cycle regenerated the starting material with each turn. Same thing here. It's a cycle. That's a little bit less important as far as the exam is concerned and for understanding the whole thing. Just like any other cycle we talk about, each one has inputs and outputs. I'm going to put an X to that because you don't necessarily need to know that. But these things you do need to know. This is good test questions. Which of the two stages uses carbon dioxide? Calvin cycle. Again, I'll remind you of the hint that I gave you in chapter 6 when we talked about respiration. For this whole exam, when you have any questions about carbon dioxide, so whether we're saying in respiration which stage produces carbon dioxide, or if we're talking about photosynthesis, which stage uses carbon dioxide, the answer will always be some sort of cycle. And the hint there is the C's, right? Calvin, cycle, carbon dioxide, right? Citric acid, cycle, carbon dioxide, it all goes together. Anyway, so yeah, that's one test question. Which of the two stages uses carbon dioxide? And the answer is Calvin cycle. And this is the part that'll trick people. I don't mean it for it to be tricky, but I can see how it's tricky. Which of the two stages uses ATP? Right? If we're talking about using ATP, the answer is the Calvin cycle. So remember, light reactions produce ATP. Calvin cycle uses it. And that should make sense because you should know at this point, because we keep talking about it. ATP is the energy source for cells. And obviously Calvin cycle is the thing doing the work. It's the thing producing the glucose. So what's the thing that's going to need the energy, right? In light reactions, as it implies in the name, that's all about the energy, right? It's all about producing energy, so to speak. That would make sense that light reactions would produce ATP and the Calvin cycle would use ATP. Again, the other test question, which of the two stages uses NADPH, right? Which of the two stages is using those high energy electrons captured by being carried around by the NADPH? And the answer is the Calvin cycle. And of course, the opposite question would be, which of the two stages produces those high energy electrons in the form of NADPH? And that would be light reactions. So here's the real kicker, the most important thing. Well, not the most important thing, but that's what we have in, right? Those are the ingredients. We have carbon dioxide, we're using ATP, we're using high energy electrons, but what are we making? And I'm gonna circle this for the sake of grading. That's what you need to know. We're making glucose. But technically, we don't go straight to glucose. Technically, Calvin cycle produces something called glyceraldehyde 3-phosphate, abbreviated as G3P. And that G3P is then used to make glucose. But I'm going to put an X to that just because I want to make things more simple for you for the exam. So as far as we're concerned, for the exam, Calvin cycle, or photosynthesis in general, produces glucose. Yes, technically it actually produces G3P, and that is then used to produce glucose. But as far as we're concerned for the exam, it is used to produce glucose. And maybe that would be a good extra credit question. I might say, it might literally say it like that. As far as we're concerned, Photosynthesis produces glucose, but what is the intermediate molecule? What molecule is it that we use to actually make the glucose? The answer would be G3P. But I probably wouldn't ask that question, because for me to ask it that way, then I'd have to literally put in writing, photosynthesis makes glucose. And that will be a test question, so I don't want one question to give away the other ones. But here's a good independent work idea. This last bullet point. That G3P, again, is used to make glucose, which is all we're concerned with for the sake of the exam. But it also makes other organic compounds. So what else is photosynthesis making? Other than G3P and glucose, what else is that G3P used for? So if you want, that's a good independent work topic. And to remind you, because some people, it's easy to do, it's a good time to remind you about something with independent work. You don't have to, like, when you look it up, you don't need to, like, give all this information. Because if you try to do that, then you run the risk of, like, rewording your source, which will give you 0 credit. Just read it, memorize it, and answer it. It could be a short answer. You could say something, this is completely not correct, but let's just say it to help you remember. You might look it up and say, okay, in class you asked what G3P makes other than glucose. I looked it up, and according to photosynthesis.com, it also makes meth. That's not the answer, but you know what I'm saying? Like, you could just keep it that simple. And of course, when you look it up, it's going to be a long article that explains how it makes it and all these different things. But please, I'm telling you, it'd be easier for you just to keep it short and sweet. Obviously, the shorter it is, the fewer points you get, but that's okay. Because if you keep turning in a bunch of short things like that, you'll be in business. Anyway, any questions about this slide? Because the next slide I'm going to show you is a picture of photosynthesis. Not a picture, a drawing of a Calvin cycle. All right, here we go. This is the Calvin cycle. Here we have the acceptor molecule. Technically, that acceptor molecule is the RUBP sugar. I'll put it next to that. You don't need to know that. You'll notice it has 2 phosphates. When you really get down to it, that is important for the process because we're making, well, you'll see. But as far as the exam is concerned, the fact that it has 2 phosphates don't matter. But anyway, we have this molecule. It hooks up with the CO2 from air. How many carbons are in carbon dioxide? 1. Good. How many carbons are in this? 1, 2, 3, 4, 5. No math majors here. We've already determined that. I've already asked you before. But can someone tell me what 5 plus 1 is?
6.
Yes. So I'm only pointing that out because if you're really studying, and I didn't explain this, then this might piss you off. It will piss me off. Because 5 plus 1 equals 6, not 3. Well, what this picture doesn't show you is that this 5 carbon molecule accepts 1 carbon from carbon dioxide and it gives us a 6 carbon molecule. And then that 6 carbon molecule is broken in half. So now we actually have 2 3 carbon molecules. That detail does not matter for the exam. I'm just saying that because, again, if you're studying and studying hard and you think about it, that would blow your mind. Like, how do we go from 5 plus 1 to 3? And that's how it happens. There's a step that's missing from there. And if you want to, you could look that up for independent work. What step is that called? When the RUBP sugar gets the carbon, what is the molecule between this and this? Also, for that matter, what is this 3 carbon molecule called? They all have names. Not that it matters. I just don't understand why they told you the name of the RUBP sugar, but not the 3 carbon molecule. Anyway, so we have that 3 carbon molecule. So we use ATP that we gather from the light reactions. We use that to change it. We don't even tell you how, but that 3 carbon molecule is changed into G3P sugar, which as far as these drawings are concerned, looks the same, right? 3 carbons and a phosphate, 3 carbons and a phosphate. Just take my word for it, they are different. We pump in some high energy electrons that again we got from the light reactions. And here, this is where it's important, the fact that we have technically 2 of these, right? Because 5 plus 1 equals 6. So we had a 6 carbon molecule, we broke it in half. Now we have 2 3 carbon molecules. This is why that's important, because at this point, we've made 2 G3P sugars, right? Because we had 2 of these. We've made 2 of these, one of which leaves and goes and makes glucose and other organic compounds. The other one stays in the cycle. The other one goes back. We do some more magic to it. We add some ATP to it, and we go from G3P sugar back to RUVP sugar, and the cycle continues. Obviously, there's a lot missing here. As you can see here, we went from 3 carbons to 5 carbons. So where did the other 2 carbons come from? That's all explained if you really, really study photosynthesis, but you don't need to know it. You can look it up for independent work if you want. There's a lot that goes on between here and here. I'll also point this out for the sake of the lab today. Each single one of these steps, including the ones that are not shown on here, requires a very specific enzyme. One enzyme for this step, a different enzyme for that step, a different enzyme for that step. So if things get too hot, the enzymes aren't going to work properly. If things get too cold, the enzymes aren't going to work properly. Here's a picture of everything. When we get back on Friday, we'll do the review. And the last word for attendance is going to be hand sanitizer. All right, you want to do the picture?
You all have been in the system. But it left.
That's all right. Well, I want to, if you don't mind, let somebody else take it just so they can get the points too. Who wants to take the picture for us? All right, thank you.
Probably yours to what they're going to do.
Yeah. No one was feeling the school spirit today.
I looked at my hoodie. Oh, I might have read.
Oh, it's gonna be a lot easier in the cooler months, because then, no matter what you're wearing underneath, you can always just wear like a sweatshirt. Thank you, Bruce. Thank you. Have a good day. You too. Don't forget, guys, come to me if you need help.
I have a question.